So here we are, the last lecture of the semester. Woohoo! So we'll be looking in this lecture at the special sense of vision and the structures of the eye that support this sense. So the eye, of course, is the organ of vision, and there are some accessory organs associated with the eye. The first is the conjunctiva. These are the mucous membranes that are found on the inner surface of the eyelid. And so it's a mucous membrane, and it helps to, the mucus that it produces helps to allow the eyelid to slide smoothly over the cornea as we close our eyes while we're blinking or while we're asleep. The lacrimal apparatus, if you recall, there was a lacrimal duct in the skull, in the facial bones. So the lacrimal apparatus is the organ that produces tears and conducts tears to the surface of the body at the eye. The lacrimal glands are found superior to the eye and slightly lateral, and they produce a watery secretion known as tears, and the lacrimal ducts transport the tears then to the eye. And tears are watery, and they include enzymes that help to kill bacteria. So we have enzymes there that are responsible for maintaining a bit of chemical protection to the body so that we get relatively few infections coming into the eye, even though the eye is directly exposed to the environment. And then finally, there are 6 extrinsic muscles associated with the eye that are responsible for moving the eye both up and down and side to side, and then all of the movements in between that. And these skeletal muscle, but very, very small motor units. So can have as few as 5 to 10 muscle fibers in a motor unit. And so that gives us great precision in being able to move the eye and and train the eye on a very specific target. And this is, vision is one of the senses that we do very, very well as humans. Our abilities compared to other mammals around smell and hearing are pretty limited, but vision is something that we do very well. And so we see that, we see that importance of vision in our evolution in this small motor unit control of the eye. So if we look at the eye itself now, what are some of the anatomical features? The eye is a structure that the eye is like a, what kind of an organism is Shrek? He's a troll? Yeah, the eye is like a troll. It has layers. So the outermost layer of the eye is the outer tunic. It is where the outer tunic is built of a white fibrous connective tissue, is the sclera. That's the white of the eye. The cornea is anterior, and it is the transparent portion of the outer tunic. So they are both connective tissues, the sclera and the cornea, but the protein fibers in the cornea are transparent. And so this is the outer protective covering, but it does allow light to enter to the interior of the eye. So light has to pass through first the cornea as it makes its way toward the retina, where we're going to interpret or receive the sensory impulse or sensory information. So, So the outer tunic, the lens is found in the interior of the eye. It's composed of very densely packed columnar epithelial cells, and these epithelial cells are filled with clear crystallized proteins. And so this is the lens is important for being able to receive the light that is passing through the cornea and through the pupil, and then focus that light toward the rear of the eyeball. So So we see that the lens will take light as it comes in and bend it and focus it directly at the rear of the eye cavity itself. So if this is a tree that we're looking at, then light wavelengths, or the light energy, light will come from somewhere. It bounces off the tree, and what we wind up projecting, thanks to the cornea, to the lens, is an upside down image of the tree at the back of the eye. I've done a terrible job of showing that. So if this is our tree here, as the light passes through the eye, so we pass through the cornea, through the pupil, and into the eye, then light coming in is bent, and it produces an image on the back side of the eye where the image is upside down, directly on the back side of the eye. Hopefully that now makes some sense. Okay, so the middle tunic of the eye is composed of two materials. There is for some reason, yeah, okay. I'm sorry, I just had to check to see where I was going. There is the ciliary body. These are the muscle fibers and the ligaments. So here's muscle fibers, here are ligaments that hold the lens in place. And so they attach to the lens capsule. So we got a capsule of connective tissue around the lens, and they attach back to the wall of the eye here at this middle layer. And these muscles here of the ciliary body, they hold the lens in tension. So the lens would normally be circular, and if you recall from the sheep eyes that we dissected, the lens was circular, and that was because there was no muscle or ligaments pulling against it. But when the ligaments and the muscles of the ciliary body pull and tug against it, then it takes on a more disc-like shape. And so they hold the lens in tension, keeping it relatively flat. But as we need to focus the lens in order to get the image to be in focus on the backside of the eye, the backside of the interior of the eye, we can relax the muscles of the ciliary body, and that allows the lens to relax a bit as well and become more round, and so we can focus on nearer objects that way by allowing the lens to become rounder. And that is referred to, that process of relaxing so that we can refocus the eye is referred to as accommodation. So there are cells associated with the ciliary body that also secrete the aqueous humor. That's the very thin, watery liquid that fills the interior of the front cavity of the eye, the anterior cavity. And so aqueous humor is found in the anterior cavity. That's everything anterior of the lens itself. So the anterior cavity is here between the cornea and the iris. So there's the anterior chamber of the anterior cavity, and there's a posterior chamber of the anterior cavity of the eye. Okay. So also part of the middle tunic is the choroid coat. The choroid coat is a layer, it's a very vascularized layer that's in between the sclera and the retina. And it has cells in it that produce really darkly pigmented melanocytes. So melanocytes, if you recall from when we were talking about skin, melanocytes are little packets of filled with melanin. And so this dark layer helps to prevent light coming in from any other location than being allowed to come in through the pupil, the opening in the iris. The iris is also part of this middle tunic, and it is the color part of the eye. And here we're looking at the iris from the back side. And so the iris has an opening in the center. Oh, wait, I'm sorry. We're looking at it from the front side because there's the lens at the back side of it. There's an opening in the center of the iris which allows light to come in toward the lens. And there are muscle fibers. There are radial fibers that, as these radial fibers constrict or contract, they pull open the iris. So contraction of the radial fibers makes the iris larger. And then to pull the iris back into a constricted shape, we have to relax the radial fibers and constrict or contract the circular fibers. Circular fibers then will make the iris, so there are fibers that are arranged around the perimeter, like arranged so that when they contract and get smaller, then the pupil becomes smaller as well. So they work in opposition to each other, circular fibers constricting the pupil, while the radial fibers dilate the pupil, make the pupil larger. And the pupil is just an area where there is no iris. So it's just this hole in the center of the iris that allows light to pass through to the lens. And then there is an inner tunic, and this is the inner lining here. I went back, so it's the inner lining here, and it's called the retina is the inner tunic. So if this is the back side of the eyeball, then light first passes through a layer of nerve fibers that all are heading toward where the optic nerve leaves the eye. And then they pass through a layer of all of the neurons that connect to the sensory receptors. So this is an interesting arrangement in the way the eye is constructed. We have to send light through these layers that actually obscure some of the light to get to the sensory receptors. The photoreceptors are at the very back side of the retina. And here you can see the choroid coat. This is where all the melanocytes are, and then the sclera behind it. So at the very back side of the retina is where we find the sensory receptors, the rods and the cones. So in the retina, there are two kind of landmarks. So if we're looking into the back side of the eye here, at the very center of the eye, at the very, like if this is the lens in the middle and here's the iris around the lens. So right at the bullseye center of that is the fovea centralis. It's a little pit. If you look at it from the side, here's the back of the retina. Fovea centralis is like this little pit in the retina. And it is found at the center of what's called the macula lutea, which is a region of yellow pigmentation. And that's right at the center of where we focus that upside-down tree. So here's the upside-down tree on the back side of the eye. And the macula lutea is right at the center part of where we focus that image. And then at the very dead center of the macula lutea is the fovea centralis. So we have scattered around the retina, we find rods and cones. But here outside of the macula lutea, it is almost entirely the rods. But here in the very center of the macula lutea, at the fovea centralis, this is a region that is densely packed with cones. And so here we get our best color vision is right there at the fovea centralis because it's nothing but cones, and because cones are really good at giving crisp, clear vision. This is the area where we have our sharpest vision. And so as we're moving our eye around looking at things, we are constantly attempting to keep the fovea centralis right at the very center of whatever it is that we're looking at, because that's where we're going to get our very best, our very sharpest, clearest vision. Okay, so here we are. This is now an image, this is a photo taken right through the iris and through the lens at the back side of the retina. So we're getting a nice view of the retina. Here is the macula is kind of right here in this area. And right in the center of it, there's the fovea centralis. And off to the side is the optic disc. It is medial, so the nose would be here. There's the nose and the nostrils. Woohoo! So here is the optic disc, and this is the region where all of those, remember, all of the nerve fibers, they're all heading toward the optic disc, bringing their axons to this point where the optic nerve exits the eye. And at this spot, there are no photoreceptors, so no rods and cones at all. And so we talk about that as the blind spot. And it's also this point at which the blood vessels that serve the eye enter and exit. So we have arteries or arterioles coming in through this optic disc and venules leaving through the optic disc. The posterior cavity of the eye is filled with a clear but much less watery, very jelly-like fluid called the vitreous humor. And there's lots of collagenous fibers in there. And so this jelly-like kind of dense fluid helps to support all the internal structures of the eye, forces the retina back against the back wall of the eye because the attachment there is very, very fragile. And it's possible with a sharp blow to the head around the eye region to rip the retina away from the choroid coat, and then vitreous humor can kind of creep behind the retina, and then it's impossible to actually focus on the retina anymore. And so a detached retina is a real problem, and if it doesn't attach itself again, then it can lead to very, very bad vision and blindness. So let's look at the pathway of light then, as light waves are reflected off of an object. So again, just because it's easy to draw, let's draw our Christmas tree, and it's the right time of the year, right? So here's our Christmas tree, got a little star at the top, got ornaments hanging all over. Okay, that's good enough. So the way that light works is that we have some light source. So here's the sun, and it sends out light rays of all colors. So, you've seen rainbows. That's where we can fragment white light into the different color spectra. So we send out all these different colored light wavelengths, and some of them are absorbed by the object that we're looking at, and some of them are reflected. So they bounce off and are reflected. So the reason that our tree looks green is because red wavelengths are absorbed, blue wavelengths are absorbed, but green wavelengths are sent back toward the eye. So these green wavelengths of light enter through the cornea. They travel through the aqueous humor that's found in the anterior chamber of the anterior cavity. They pass through the hole in the iris, the pupil. The wavelengths then pass through the aqueous humor that's found in the posterior chamber of the anterior cavity. The light wavelengths pass through the epithelial cells of the lens, and then they pass through the vitreous humor that's found in the posterior cavity, and they're refracted is that term where we bend light. Wavelengths pass through, they're refracted, and we wind up focusing our tree here on the back side of the eye with the center of our vision right here at the macula lutea, with the very center right there at the fovea centralis. And so those wavelengths will then stimulate the rods and the cones of the eye. And if there's sufficient stimulation, then we'll get action potentials that are transmitted through those through the sensory neurons, and they bring their action potentials up the optic nerve toward the visual cortex of the occipital lobe of the brain. Okay, so let's talk now about the specialized photoreceptors that are there at the back side of the retina. Rods are by far the most numerous of the photoreceptors. We have about 100 million of them placed throughout the retina. They are sensitive not to wavelengths of light, but the amount of light stimulation that's being received. And so it produces our black and white vision, where there are a greater number of of photons of light striking the retina. Then we produce white appearance. Everything gets a light-colored appearance or light appearance. Where there's few of them, then everything is darker. And they are most active in dim light. In fact, in very bright light, they tend to become inactive. And we'll talk about that in just a minute. And they don't produce a very sharp image. The rods don't. They instead produce a kind of fuzzy image, because these rods, multiple rods here, will submit information along this neuron, and then, this one's also submitting along this neuron, which is, so there's a lot of overlap in rod transmission of information. And so we get multiple rods that are all then connected back to this single sensory nerve fiber through these sensory neurons. So there's a lot of information being mushed together into a single, and yes, mush is a anatomical term, certainly. But we mush a lot of information together down a single nerve fiber, and so that gives us a somewhat fuzzy image based on rods alone. So rods, black and white vision, kind of fuzzy images because the edges, the margins of things are not clearly laid out, and they're most active in dim light. They become inactive in very bright light. And so this is why at nighttime our vision gets a little blurry, is because it's mostly the rods that are being stimulated. Cones are much less numerous, only about 3,000 versus 3 million versus 100 million. And they are not so sensitive to the amount of light received. Instead, what they pay attention to is the wavelength of light that's received, which color light is being transmitted. They provide very sharp images because very few cones transmit via a single axon. So in this picture, we're seeing that each one of these cones has its own axon that's going to contribute information to the occipital lobe. Again, most abundant in the fovea centralis, which provides only color vision. And so we get good sharp images because of this distinction between cones and the nerve fibers that they're transmitting along. And we get color vision because cones are capable of responding to different wavelengths of light. So now let's talk about how the rods and the cones actually work. So rods work because they produce this purplish kind of pigment called rhodopsin. And when light energy lands on the pigment in the rods, it breaks the rhodopsin apart into two molecules. One is a colorless molecule called opsin, and the other is a yellowish pigment called retinol. So we break rhodopsin apart into opsin and retinol. Retinol is, by the way, a product of, we use vitamin A to make retinol. So when opsin is available in the rod cytoplasm, that opens up sodium channels. It closes sodium channels that are opened. And so this results in hyperpolarization of the membrane of the rod and the release of neurotransmitters that cause either excitation or inhibition at the associated neurons. It depends on the neurons, or on the neurotransmitter that's being released. So it's the presence of a material, opsin, that's made when light hits the rod that causes impulses, nervous impulses, to be produced. So when we're out in really bright light, rhodopsin is being degraded in the rods more quickly into opsin and retinol more quickly than it can be made. And so all we get is a lot of static information coming from the rods. There's not much information that comes. It's so this is why when you're out in bright light and then go into a really dark room where there isn't a lot of color light coming, and the best we can do is discriminate between lots of light coming in and very little light coming in, then we don't have very good vision for a while. It takes some time for us to build up again levels of rhodopsin that will allow us to be able to see. So when we become dark adapted, though, then we can make rhodopsin more quickly than it's being degraded. And so we're capable then of perceiving shades of gray. And we can increase our rod sensitivity by up to 100,000 times versus when we are out in bright light. So the rods are very sensitive in dark light or darker conditions. And that's because there's plenty of rhodopsin around to kind of to shut down or open up those sodium channels so that we're getting the appropriate excitation or inhibition. Okay, so lots of rhodopsin, then we can perceive more easily very low levels of light entering the eye. So on the other hand, okay, so we said that if you're light adapted and move into a dark space, you don't have very good vision. Well, if you're dark adapted and move out into really bright light, the process of degrading or bleaching this rhodopsin very, very quickly like that triggers pain impulses to be produced, and so it's really painful, and I think all of us have had that experience of being in the movie theater for a matinee and coming out to bright light, and wow, it just hurts to even look at look around because of all of the rhodopsin that's being bleached and the pain impulses that will trigger. Okay, cones, on the other hand, come in one of three flavors. They produce not rhodopsin, but erythrolabe, chlorolabe, or cyanolabe. Erythro means red. So these are cones that are able to absorb and perceive or create sensations when red wavelengths of light impinge upon them, upon that cone. Chlorophyll is a green pigment, so chloro means green. Chlorolabe is capable of responding to green wavelengths of light. Cyanolabe, cyan means blue. These are cones that respond to blue wavelengths of light. So, again, light comes from the sun and from light sources in our house with multiple wavelengths all at one time. But when you look at light, when you look at I'm looking up right now at the now I can't think of what kind of light bulbs they are. The fluorescent light bulbs that are here in this room where I'm doing this lecture. I look up and what I see is white light. And that's because I'm getting wavelengths of red, green, and blue in relatively equal proportions. So if we sense all three of these wavelengths at the same time, what we get as a perception from the sensory cortex of the occipital lobe is the color white. If, on the other hand, we're in a situation where no light, none of the three wavelengths of light are being sensed, then what we perceive is the color black. So if you're looking at a black piece of paper or at a black something in your house as you're listening to this, the reason that it appears black is because there is no light being reflected from the pigments in whatever that surface is. So when there's no light reflected back from an object, then we get the perception black, when there is all three wavelengths being projected or reflected back from an object, like looking at a white piece of paper, then we see this color white. If we see just red and blue, then wavelengths, then we perceive the color purple. If we see red and green wavelengths, then we perceive the color brown. If we see green and blue, then we get the all the colors that are in between green and blue. So so it's depending on the wavelength that we get, we sense all of the colors of the spectrum. So it's the combinations of these wavelengths that give us the colors of a spectrum. So they also sense the amount of light that is coming at them, and that's when they use the pigment iodopsin that also, like rhodopsin does, bleaches in response to light intensity. Okay, so a couple more things to talk about with vision. The book goes on to talk about farsightedness and short-sightedness. So be sure to read those sections. Just because I'm going to skip over them doesn't mean that they're not important. So be sure to take a look at those. But there's a couple more things that I wanted to make sure that you're completely aware of around this. And one of them is that because we have two eyes and they're spaced slightly apart from each other, each of our eyes receives a slightly different image at the retina. Things are spaced over just a little bit, or we get slightly different angle on whatever it is that we're looking at. And so the brain is capable of integrating these slightly different images. So the information coming from the right eye is decussate. It moves over to the left occipital lobe. The information coming from the left eye is decussate and moves over to the right occipital lobe. And we take those two images in the occipital lobe and integrate them. And this allows us depth perception, as well as being able to perceive height and width of things, because we have these two images that are slightly different from us and from each other. And we talk about that as stereoscopic vision. And so understand that it's the difference between the two images that are coming from the different eyes that allow us to get that perception of depth to things, and length to things, and width to things. And last of all, the visual nerve pathways, the axons that are coming from the ganglion cells in the retina, they merge, they form the optic nerve, and some of those fibers are decussate at the optic chiasma, it's called. So this chiasma, the letter chi is the Greek for the letter X, and hopefully you saw on your sheep brain where the optic chiasma is. And so those fibers that are decussate then travel to the thalamus and then move onward to the occipital lobes for interpretation. And then some of them do stay ipsilateral, so stay on the same side, but many of them are decussate as well. Okay, so there it is. Everything you ever wanted to know about the eye, but were afraid to ask. Be sure again to read this stuff about myopia and hyperopia. That's nearsighted and short-sightedness and farsightedness. The lifespan changes on your own. Make sure that you understand those concepts comfortably as well. All right, best of luck to you. And as always, we've got 2 days now. I will be doing office hours as usual on Tuesday, so right at lunchtime, but I'm teaching. I will be in room 210, and I'll be in my office. Those are the two places you'll be likely to find me. But I'll be on campus all day on Tuesday, and then on Wednesday I'll be on campus from about noon until class time. So you can always find me before class as well. All right, best of luck to you, and I'll see you on Wednesday.